This is Chavez from the Food Technology Department, and the topic is foodborne infections. Foodborne infections are caused by the entrance of pathogenic microorganisms contaminating food into the body and the reaction of the body tissues to their presence. These can be either fungal, bacterial, viral, or parasitic. Foodborne infections tend to have a long incubation periods and are usually characterized by fever. There are several types of foodborne infection such as bacterial, meiotic, or fever. Bacterial foodborne infections include cholera, salmonellosis, typhoid fever, shigellosis, yersiniosis, Escherichia coli infection, Campylobacteriosis, Vibrio paramolytis, listeriosis. Meiotic foodborne infections include Candida species, Sporthrix species, Vangella species, etc. Viral foodborne infections include hepatitis A, Norwalk virus, and poliomyelitis virus. Salmonellosis. The Salmonella constitute a group of organisms with over 2,000 different serotypes. These organisms are capable of causing disease in animals and man when taken into the body in sufficient numbers. Many Salmonella species have a wide host range. These are the organisms which commonly cause food poisoning. The control measures for salmonellosis are efficient refrigeration, hygienic handling of food, consumption of properly cooked meat, complete thawing of frozen meat, and adequate cooking. Heat processing of milk, meat, fish, poultry to destroy salmonella organism in the food. Typhoid and paratyphoid fever. Enteric fever include typhoid and paratyphoid fevers caused by Salmonella typhi, Salmonella paratyphi A, B, and C respectively. The serotypes are similar to other Salmonella bacteria, but unlike them, they are essential parasites of man. S. typhi possesses capsular antigen in addition to the usual O and H found in the other serotypes. The control measures for typhoid and paratyphoid fevers are hygienic control of food and water supplies, detection and treatment of chronic carriers, vaccination using TAB vaccine. The vaccine contains a mixed culture of S. typhi and S. paratyphi. The vaccine protects for 5 to 7 years. Camelobacteriosis. Camelobacter are a group of tiny, strictly micro-aerophilic curved or spiral gram-negative rods. Campylobacter jejuni and Camelobacter coli cause food poisoning and are associated with acute enterocolitis in man. Campylobacter jejuni occurs in large number in cattle feces and poultry as normal flora. Camelobacter coli are commonly associated with human diarrhea and enteritis in pigs, mostly in association with Treponema hydrocenteriae. Preventive measures for camelobacteriosis are thorough cooking of all food stuffs derived from animal sources, prevention of recontamination after cooking, proper refrigeration of food, recognition, control and prevention of Camelobacter infections in animals, and maintenance of high standard of hygiene. Escherichia coli foodborne infection. Escherichia coli are potential food poisoning pathogens which are widely distributed in low numbers in food environments. E. coli strains involved in foodborne infections fall into the following groups: enteropathogenic E. coli, EPEC, enterotoxigenic E. coli, ETEC, enteroinvasive E. coli, EIEC, and enterohemorrhagic E. coli, EHEC. The control measure for Escherichia coli foodborne infections are proper cooking of hamburger and other meats, avoidance of cross-contamination of food in the kitchen, and good personal hygiene. Shigellosis. Shigellosis is caused by members of genus Shigella. The species involved includes Shigella dysentery, Shigella flexneri, Shigella bodii, Shigella dawsoni. All strains of Shigella possesses potent exotoxins which are carbohydrate lipid protein complexes. The infective dose is smaller than that of Salmonella except S. typhi. The preventive measures for shigellosis are practice good hygiene and sanitation. Wash hands well with water and soap each time you use the ablution. The home and surroundings should be kept clean to prevent contamination of food and water supply. Proper disposal of human waste or sewage. Cholera. Cholera is caused by Vibrio cholera bacterium. Cholera vibrios are ingested in drink of food. In natural infection, the dosage is usually very small. The organisms multiply in a small intestine to produce a very potent enterotoxin, which stimulates a persistent outpouring of isotonic fluid by the gut mucous cells. The control measures for cholera are provision of potable water, proper sewage disposal, proper cooking and hygienic handling of food, observation of personal hygiene, vaccination. The heat-killed phenol-preserved vaccine has prevention and protection that last for 3 to 6 months. Thank you.